So this is a talk about gene drives. I'm going to start by telling you a brief story. 20 years ago, a biologist named Anthony James got obsessed with the idea of making mosquitoes that didn't transmit malaria. It was a great idea, and pretty much a complete failure. For one thing, it turned out to be really hard to make a malaria-resistant mosquito. James managed it finally, just a few years ago, by adding some genes that make it impossible for the malaria parasite to survive inside the mosquito. But that just created another problem. Now that you've got a malaria-resistant mosquito, how do you get it to replace all the malaria-carrying mosquitoes? There were a couple options, but plan A was basically to breed up a bunch of the new genetically engineered mosquitoes, release them into the wild, and hope that they pass on their genes. The problem was that you'd have to release literally 10 times the number of native mosquitoes for that to work. So in a village with 10,000 mosquitoes, you'd release an extra 100,000. As you might guess, this was not a very popular strategy with the villagers. Then, last January, Anthony James got an email from a biologist named Ethan Beer. Beer said that he and his grad student, Valentino Gantz, had stumbled on a tool that could not only guarantee that a particular genetic trait would be inherited, but that would spread incredibly quickly. If they were right, it would basically solve the problem that James had been working on for 20 years. As a test, they engineered 2 mosquitoes to carry the anti-malaria gene, and also this new tool, a gene drive, which I'll explain in a minute. Finally, they set it up so that any mosquitoes that inherited the anti-malaria gene wouldn't have the usual white eyes, but would instead have red eyes. That was pretty much just for convenience, so they could tell at a glance which was which. So they took their 2 anti-malarial red-eyed mosquitoes, put them in a box with 30 ordinary white-eyed ones, and let them breed. In 2 generations, those had produced 3,800 grandchildren. That is not the surprising part. This is the surprising part. Given that you started with just 2 red-eyed mosquitoes and 30 white-eyed ones, you'd expect mostly white-eyed descendants. Instead, when James opened the box, all 3,800 mosquitoes had red eyes. When I asked Ethan Beer about this moment, he became so excited that he was literally shouting into the phone. That's because getting only red-eyed mosquitoes violates a rule that is the absolute cornerstone of biology, Mendelian genetics. I'll keep this quick, but Mendelian genetics says that when a male and female mate, their baby inherits half of its DNA from each parent. So if our original mosquito was AA and our new mosquito was AB, where B is the anti-malaria gene, the babies should come out in 4 permutations: AA, AB, AA, BA. Instead, with the new gene drive, they all came out AB. Biologically, that shouldn't even be possible. So what happened? The first thing that happened was the arrival of a gene editing tool known as CRISPR in 2012. Many of you have probably heard about CRISPR, so I'll just say briefly that CRISPR is a tool that allows researchers to edit genes very precisely and easily and quickly. It does this by harnessing a mechanism that already existed in bacteria. Basically, there's a protein that acts like a scissors and cuts the DNA, and there's an RNA molecule that directs the scissors to any point on the genome you want. Protein that acts like a scissors and cuts the DNA, and there's an RNA molecule that directs the scissors to any point on the genome you want. And there's an RNA molecule that directs the scissors to any point on the genome you want. The result is basically a word processor for genes. You can take an entire gene out, put one in, or even edit just a single letter within a gene. And you can do it in nearly any species. Okay, remember how I said that gene drives originally had 2 problems? The first was that it was hard to engineer a mosquito to be malaria resistant. That's basically gone now, thanks to CRISPR. But the other problem was logistical. How do you get your trait to spread? This is where it gets clever. A couple of years ago, a biologist at Harvard named Kevin Esvelt wondered what would happen if you made it so that CRISPR inserted not only your new gene, but also the machinery that does the cutting and pasting. In other words, what if CRISPR also copied and pasted itself? You'd end up with a perpetual motion machine for gene editing. And that's exactly what happened. This CRISPR gene drive that Esvelt created not only guarantees that a trait will get passed on, but if it's used in the germline cells, it will automatically copy and paste your new gene into both chromosomes of every single individual. It's like a global search and replace. Or in science terms, it makes a heterozygous trait homozygous. So, what does this mean? For one thing, it means that we have a very powerful, but also somewhat alarming new tool. Up until now, the fact that gene drives didn't work very well was actually kind of a relief. Normally, when we mess around with an organism's genes, we make that thing less evolutionarily fit. So biologists can make all the mutant fruit flies they want without worrying about it. If some escape, natural selection just takes care of them. What's remarkable and powerful and frightening about gene drives is that that will no longer be true. Assuming that your trait does not have a big evolutionary handicap, like a mosquito that can't fly, the CRISPR-based gene drive will spread the change relentlessly until it is in every single individual in the population.
We have individuals who are religious fundamentalists, religious fanatics, and this is the expression, RT-PCR, real-time PCR expression of the VMAT2 gene. Over here, we have individuals. So let me complete. So over here, we have individuals who are not particularly fundamentalist, not particularly religious, and you can see there's a much reduced expression of this particular gene, the VMAT2 gene. Another evidence that supports our hypothesis for the development of this approach.
What you see here is by spreading this virus, we're going to eliminate individuals from donning on a bomb vest and going into a market and blowing up the market.
So our hypothesis is that these are fanatical people, that they have overexpression of the VMAT2 gene, and that by vaccinating them against this, we'll eliminate this behavior. So we have some very, very remarkable data in this next slide. Here we have two brain scans. These are fMRIs. These are two different individuals with different levels of expression of VMAT2. On top is an individual who's a religious fanatic, an individual, and we've repeated this numerous times, that has high levels of VMAT2. Now, this individual down here who had low levels of the VMAT2 gene, this individual would self-describe as not particularly religious. In each case, these individuals were read a religious text. This individual lit up the right middle frontal gyrus, shown here, and that's a part of the brain that's associated with theory of mind. It's a part of the brain that has to do with intents and beliefs and desires. In marked contrast, here's an individual who would not particularly self-describe as religious, and when they're read a religious text, what you see is that this part of the brain called the anterior insula lights up. This is a part of the brain that's associated with disgust or displeasure on hearing something.
So you're suggesting I take a CT scan with me when I'm evaluating people to determine whether I put a bullet in their head?
So the data that I'm presenting here supports the concept that we're proposing, and I think that we would not propose to do CT scans or fMRIs on individuals out in the hinterlands of Afghanistan. The virus would immunize against this VMAT2 gene, and that would have the effect that you see here, which is it's essentially to turn a fanatic into a normal person. And we think that will have major effects in the Middle East.
How would you suggest that this is going to be dispersed, the aerosol?
Well, so the present plan and the tests that we've done so far have used respiratory viruses, such as flu or rhinoviruses, and we believe that that's a satisfactory way to get the exposure of the largest part of the population. Most of us, of course, have been exposed to both of those viruses, and we're quite confident that this will be a very successful approach.
This is fascinating. What's the name of this proposal?
Yeah, so the name of this project is FunVax, which is the vaccine for religious fundamentalism.
And you have a proposal already?
The proposal has just been submitted, and I think that the data that I have shown you today would support the development of this project, and we think it has great promise.
Stand therefore, having your loins girt about with truth, and having on the breastplate of righteousness, and your feet shod with the preparation of the gospel of peace. Above all, taking the shield of faith, wherewith ye shall be able to quench all the fiery darts of the wicked, and take the helmet of salvation, and the sword of the Spirit, which is the word of God, praying always with all prayer and supplication in the Spirit, and watching thereunto with all perseverance and supplication for all saints,